Welcome to Lecture 3 of the GMP and Quality Fundamentals series. In Lecture 2, we discuss GMP principles, standard operating procedures, documentation practices, deviations, and audits. In this lecture, we move into the laboratory environment. We will focus on testing, analytical methods, data, and results. This is the stage where quality control becomes practical and highly visible. Why do we test products? Testing exists to answer one essential and critical question. Is the product safe and compliant? Products are tested to protect consumers, to meet regulatory requirements, and to ensure consistent quality from batch to batch. Testing is not performed because we enjoy generating data. Testing is performed because important decisions depend on reliable data. What does a quality control laboratory do? A quality control laboratory analyzes raw materials, in-process samples, and finished products. Typical QC activities include sampling, chemical testing, microbiological testing, and reviewing analytical data. Based on laboratory results, materials and products are either released or rejected. The laboratory plays a central role in supporting these decisions. Let us consider a simple example. Imagine a cake produced in large quantities. It may look perfect, smell good, and taste fine. However, without laboratory testing, we cannot be sure that it is safe. Laboratory testing of this cake may include microbiological testing for bacteria or mold, chemical testing to verify compositions such as sugar, fat, or protein content, and testing for possible contaminants. Appearance alone is not enough to guarantee safety. Laboratory testing can generally be divided into three main categories. Chemical testing focuses on identity, assay, and impurities. Identity confirms that the substance is exactly what it claims to be. Assay determines how much of a substance is present. Impurity testing ensures that contaminants remain within acceptable limits. Microbiological testing focuses on total viable count, specific pathogens, yeasts, and molds. These tests ensure that the product does not contain harmful microorganisms. Physical testing evaluates properties such as appearance, weight, pH, viscosity, and moisture content. These characteristics help ensure product consistency and stability. What is an analytical method? An analytical method is a defined and structured procedure used to measure a specific parameter. Methods must be suitable, reliable, and validated before routine use. Examples include high-performance liquid chromatography for vitamins or active ingredients, gas chromatography, mass spectrometry for pesticides or residual solvents, and inductively coupled plasma mass spectrometry for heavy metals. The analytical method directly influences the quality of the data produced. Before an analytical method is used routinely, it must undergo validation. Method validation demonstrates that the method is accurate, precise, specific, linear, and sensitive enough for its intended purpose. Validation also establishes important limits such as the limit of detection and the limit of quantification. Without proper validation, analytical results cannot be trusted. The limit of detection is the lowest amount of a substance that can be detected, but not necessarily quantified accurately. The limit of quantification is the lowest amount that can be measured reliably with acceptable accuracy and precision. For clean label or high purity products, very low limits of quantification are especially important. Detecting a contaminant is not sufficient. It must be quantified reliably to support quality decisions. One real-life challenge in laboratory testing is the matrix effect. Samples are not pure water. They contain many components. The matrix refers to everything in the sample except the target analyte. For example, fat in a cake, sugar in supplements, or proteins in food products may influence the analytical signal. Matrix effects can distort results and lead to inaccuracies. A false high result means the measured value appears higher than it actually is. A false low result means the measured value appears lower than reality. Both situations are problematic because quality decisions are based on these results. Another challenge is carryover. Carryover occurs when residues from a previous sample contaminate the next sample. This is especially critical in trace-level analysis. Proper cleaning procedures, method design, and system suitability checks are used to minimize this risk. Quality control does not only generate data. It also reviews and evaluates data. Data review includes checking system suitability, identifying unusual results, verifying calculations, and ensuring complete traceability. Data integrity is as important as the analytical measurement itself. Many laboratory tests are based on official reference methods. These may include methods from the European Pharmacopoeia, ISO standards, or validated in-house procedures. Using recognized and standardized methods increases reliability, consistency, and regulatory compliance. ISO IEC 17025 defines general requirements for the competence of testing laboratories. It covers technical competence, method validation, equipment calibration, staff qualification, and data integrity. Accredited laboratories are expected to produce reliable and defensible results. Returning to our cake example, imagine that the product passes microbiological, chemical, and physical testing. The quality control laboratory reviews the data. Quality assurance confirms compliance with specifications. Regulatory affairs ensures that legal requirements are fulfilled. Only after all these steps is the product released. The key message of this lecture is simple. Testing supports quality decisions. Analytical methods are just as important as the results they generate. Poor data leads to poor decisions. Therefore, quality control carries significant responsibility within the GMP system. In the next lecture, we will focus on risk management, out-of-specification results, and investigation processes. Stay tuned for Lecture 4.